Okay, well, hi everyone. Thanks once again for joining us to hear about the role of proteomics in understanding the coronavirus. Today we have three speakers here to tell us about ongoing work in this area. Professors Andrea Sinz, Max Crispin and Perdi Barran. By now, I'm sure most of you know the drill, but for anyone new joining us, we are using our Slack channel for questions and discussions. Joining this using the link on the screen in front of you should take less than about a minute, so you can almost certainly have joined that before I finish doing my introductory ramble. Slack basically allows us to prioritise questions others want to hear the answers to. Use the thumbs up to let us know questions that other people have asked that you might like to hear the answer to as well. Slack also gives us the opportunity for the speakers to answer any questions towards the end of the meeting after they're done with their talks. For those needing an attendance certificate for this webinar, there'll be details towards the end of the three talks. We will also be providing another quick survey just to get some feedback again on how the meeting's going and how people are finding it. Thanks very much to those who gave us feedback last time, very, very useful. So once again, we'd like to say a big thank you to the European Proteomics Association, to the British Society for Proteome Research, to the Young Proteomics Investigators Club, and the London Proteomics Discussion Group Committee for all their help in supporting and setting up this webinar. Thanks also to the London Biological Mass Spectrometry Discussion Group, London Metabolomics Network and the News in Proteomics Research blog for promoting this event. We're also grateful for Imperial College London for providing us with webinar support. And a huge thank you to the speakers today for their time. So we are pleased to announce that our next webinar in this series will be on Friday the 29th of May, again at 2 p.m. British Summer Time. And this will feature Professor Ray Cruz-Isles talking about his work detecting SARS-CoV-2 viral envelope proteins with MALDI-TOF, and Dr. Martin Daines on his development of an MRM assay for SARS-CoV-2. So our first speaker today will be providing us with a brief overview of how a mass spectrometry community is coming together to help in the current outbreak. Professor Perdi Barran, who is based at the University of Manchester, is one of the founding members of the COVID-19 Mass Spectrometry Coalition and is involved in the management of the consortia. And before I over-introduce, I will hand over to her.
I think you can hear me now.
Sounds good.
Great. So thanks very much, Harry, for that great introduction, and thanks to the other two speakers for giving me a tiny bit of their talk time to tell you about an initiative, which actually the research that both of them have been doing has been really inspiring for what we want to do. So we have, like many of you, I think, been absolutely taken aback at how vulnerable we all are. And my initial thoughts were, we're vulnerable, I'm a measurement scientist, I've got really sensitive, really accurate instrumentation that I spend all of my time using to make reliable measurements on biological species. So what can I do to try and prevent this vulnerability? And so that was my initial motivation. And then I started talking to friends and to colleagues about what we could do. And from that initial motivation of saying, what can mass spectrometry do? And then what can we do? We formed the COVID-19 Mass Spectrometry Coalition. And our principal aim is to use mass spectrometry to reduce the harm caused by the SARS-CoV-2 virus. So there's been many, many people involved in this, and I just highlight some of the UK members here. We've also had huge support from many national societies, and in the UK, the British Mass Spectrometry Society, CAMS, the Community for Analytical and Measurement Science, and also lots of instrument manufacturers. So you can find out more about what we're doing by looking at our website, but I'll just give you a flavour now of some of the things. So we talked with many people, including Andrea, one of the speakers today, very early on. And in March, we decided to form a coalition of mass spectrometry labs, which would have national points of contact, so that each of the countries involved would be able to report back some common outputs to this coalition. And we also agreed that we would share as much as possible the methods that we use and the protocols that we use and all of the data we produce. And that's really critical, because that data piece will allow many more researchers and clinicians to do something with what we're gaining. So this is like a new way of working. It's about not waiting till you've got the best, highest impact paper, but actually get that data out there. And many groups have done that using preprint servers and other channels. So in terms of the science that we are aiming to do, and certainly the talks today and in 2 weeks' time we'll cover some of this, is to measure the response of the host, of the individual, to the infection using primarily serum, but also other biofluids. We also believe, and are starting to do this, that we can assay quality control compounds that are being used in serological testing kits and in being used in understanding the immune response. More in a more detailed way, we aim to use mass spectrometry to help with vaccine and therapeutic developments, and I'll talk about what I mean by that shortly, and to take these methods and develop them at pace to determine the prognostic features of this disease, to develop robust assays with biomarkers to help to be able to determine what response of a given individual would be. We also think that mass spectrometry has a very good role to play in developing methods to determine the lifetime of infected particles in the environment, and that's something that I know is of great interest to many people, and we think some of the particularly ambient ionization techniques could be really useful here. So from small beginnings, the movement snowballed. Everyone is at home and willing, right? So we've signed up now more than 400 members, and I invite any of you that are not signed up to join. Our membership is spread over at least 18 countries and represents more than 200 laboratories and companies and national measurement laboratories. We've got a web presence I've mentioned, and critically, we've connected to the COVID-19 EU data portal. We, with our website, will catalogue all the data sets that we can that are taken using mass spectrometers on COVID-19. We've developed methods for sample collection, and I think many of you will know the OSARIC protocol, but that's the WHO standard, and that determines how and when you can take samples from patients in hospitals. We've also developed methods for processing the samples, and many brains have worked really hard to think of ways that are practical in terms of how we can take biofluids, blood, sputum, saliva, and Andrea will talk about that, from patients in real situations and use those to do mass spectrometry on them. And I think it's critical that as much as possible we share those methods. We've applied for some funding. We don't know about that yet, and we've also applied for funding for proof of concept work with clinical samples, and we've been able to obtain ethical consent here in the UK to obtain 500 patient samples, and collection is already ongoing for that. We've linked with the EU data portal as mentioned, but we've also linked with the consortia for protein production for COVID-19, whereby a number of labs funded by Wellcome are making recombinant forms of the viral protein. And we will be using mass spectrometry methods to determine the differences between those proteins, particularly at the glycan level, which is so critical for how these proteins behave. And of course, different recombinant forms will have different glycan profiles, which will of course be different from those in the virus. And that's a piece that we really need to look at. So this is the beautiful clip art diagram I made to show what has to happen in these stages, and we send this to all of the national points of contact. We need to get samples from hospitals and perhaps from the community also, and there it's really critical that we liaise with clinicians and consider the research ethics around getting those samples. We worked really hard on sample protocols, and Andrew Pitt and Claire Ayres and many others have really been working a lot on this. Once we start to acquire samples, we of course are at the behest of the instrumentation we have, but we've really worked very hard to try and say that we will have as many common workflows as possible there, and data processing as well. Our data curation approach will be to put all the data on open servers on the PRIDE and other community standard databases. But we, as mentioned, will keep a repository, keep a catalogue of all those data sets, so that people will be able to see all of the data and search it based on proteomic investigations on COVID serum, and that could be from Brazil or UK or Belgium. You'll be able to see it all in one place. It's been an awful lot of coordination to these tracks, and the people who've been involved in the UK are mentioned on this slide. This is showing our strategy for determination of host response and disease stratification. This has been really led by Andy Pitt, Claire Mills at Manchester, Drupa Trivedi, with a lot of input from other people. We're collecting from Surrey, and Melanie Bailey at Surrey has been actually using her team in the hospital to collect samples from patients. Samples are then processed in the clinical lab, and some are taken, each patient that we sample from, we're doing the immunochemistry on that same sample. Viruses are being inactivated and then transferred to the analysis labs all over the UK, deliberately geographically spread so that we can, in the end, be close to the NHS labs that we will serve. We decided that a multi-omics approach would be really appropriate, and so we'll be doing metabolomics, lipidomics, proteomics, and in some cases glycomics, particularly on the host, on the viral antigens and on the antibodies produced. And as mentioned, all of this data will be made available. The part that is probably closest to a lot of the research that I do in my group is to map the SARS-CoV-2 viral protein dynamics and interactions. So this is an image, and there have been many structures taken of the SARS-CoV-2 spike glycoprotein, and they're fantastic. But of course, they are, in the majority of cases, in fact, I think in all of the cases, taken from recombinant forms. And recombinant forms will not have the same glycans as the host protein, as mentioned. So our efforts here are to take those recombinant forms using the work of the CPPC and map both the dynamics of the spike protein, which is a large protein and acts as a trimer, but also the way in which it interacts with both the receptor and other key protein partners. And we're going to do this in 3 university labs, so we're starting on this in the next couple of weeks, and also at LGC and the National Measurement Institute. Understanding this behaviour is really critical for vaccine development and mechanism, as I'm sure Max will describe later on. And finally, the biggest output, I think, and the one that I really invite you all to take part in is this data piece. Of course, we have many great databases, community standard databases, PRIBE and metabolites and so on, and that's where we should deposit our data. But we will be cataloguing all of those data sets to allow people to be able to search those data sets and to do this type of analysis. We will do it on our data, but everyone will be able to do it. And it's in doing that data sharing that I think we will have the biggest impact by being open and pushing it out there for the community, and of course by integrating with other data efforts. So that's the end of what we're doing. As I said, I very much invite you to join if you haven't already, to email me, or through our website. We have an administrative email, although that just comes to cost us the Lastinos and me, and to have you get involved too. So thanks very much, and I'm looking forward to the next 2 talks. I'm happy to take questions.
Okay, thank you ever so much. It's really good stuff. Really admirable effort on the part of the consortia. I think on time constraints, we're going to move on to the next talk. If anyone does have any questions, though, feel free to put them in the Slack channel. We'll come back to potentially those at the end if we get the opportunity to. But thanks again, Perdita. That's really lovely stuff. So next up, we have Professor Max Crispin. Max joined Biological Sciences at the University of Southampton from the Glycobiology Institute at the University of Oxford. He runs the Glycoprotein Therapeutic Laboratory, which is predominantly focused on developing antiviral vaccines and new antibody-based therapies against cancer. And with that, I'll hand over to you, Max.
Thank you so much, and thanks for the invitation to share our work. So we've been very interested in the structure of a wide range of viral glycoproteins, and in this slide shows just how they compare. And the immediate thing from this shared class of viruses is just how extensively glycosylated they are. And one thing that should be sort of weigh heavily on your mind is that, generally speaking, these proteins are much more glycosylated than your typical host glycoprotein. And so you can immediately think of what selective pressures must be occurring on these viruses in order. I hope everybody can hear me. I'm just
yeah, we can.
Yep. So I have my little unmute icon appear. So you can immediately think about the selective pressures that are occurring on the viruses in order for them to gain such extensive glycosylation. Now, key among these, which is easy to forget with all the exciting interactions they do, is the role in just the assembly, the role in the folding within the ER through the calnexin calreticulin folding checkpoint. So that's certainly a role because they've also got extensive disulfide bond formation, so they're very chaperone dependent. But in addition, they also influence considerable areas of pathobiology, whether that be interaction with cell surface lectins, part of that being interaction with the immune system, but also the role of glycans in immune evasion and the selective pressure that occurs there. So clearly there's considerable interest in the actual virus, but there's also interest in recombinant mimics of those viruses, which is the area which I'm going to be focusing on here. Now, why are we interested in the glycosylation recombinant mimics? Well, glycosylation processing is quite sensitive to immunogen integrity. So misfolded or aberrant protein backbones that can be revealed by the signatures within the sugars. It also influences immunogen trafficking, presentation of glycan-based epitopes, and also it can reveal the sort of degree of immunological shielding. Just how clustered are these? Just focusing on two of those, amazingly in SARS, there's already been an example in the bioarchive of a cryo-EM structure of an antibody actually forming a protein carbohydrate mixed epitope. Now in HIV, we see these all the time occurring after years of infection, mainly because of the tolerance mechanisms that glycans have. So it's quite hard to elicit antibodies which have a considerable glycan component to their epitopes. But here you can see one targeting, yes, definitely protein, but it's incorporated a fucose. And I've just included just really advert for some wonderful work by Darryl Irvin showing how the mannose content of immunogens can actually influence trafficking of the immunogen, and ultimately that is integral to how the immune system actually responds. So, two really clear interests why we're focusing. Now, we have some lessons from HIV, and here is an example of just UPLC, so fluorescent labeled release glycans of fragments of the HIV viral spike. And what's quite one thing I should just emphasize here is that we have green is the oligomannose glycans. Now these are occurring in the ER, and we're colouring magenta just the complex glycans. And of course, what we can see is that HIV has these under-processed ER-like oligomannose glycans, and I'll go on to talk about how they arise. But with this figure should really demonstrate that you can take fragments or misfolded trimers and the mannose signature dramatically changes, and that's a really important signature of correctly folded, and we've shown the EM alongside here from Andrew Ward's lab. Now you can do site-specific analysis of these, and here we're doing LCMS analysis using a fusion with proteometric software for quantitation, and we can actually measure the amount of oligomannose in these different formats and compare how they change in matched expression, all in the same recombinant system. And we can, here's plotted a sort of percentage point impact of monomer versus trimerization, and you can see in red the increase in oligomannose glycans occurring at the interface of the trimer, and similarly sort of aberrantly formed pseudotrimers where you're forcing them to trimerize, but they're not in the native configuration. You see again only a slight elevation in mannose, but not the same as the native trimer. Now, this is really important. I've mentioned about their interaction with lectins, and this just shows graphically just the different range of glycan recognition by endogenous lectins and antibodies in the case of HIV, illustrated here. But I want you to just compare the M-type lectin, this is the ER-monosidose on the left, with the C-type lectins, and you'll see that the recognition is really quite different. Enzymes really have to grab all the way around a glycan, whereas the C-type lectins can recognize on the edge. And this does solve a paradox in the sense that you can have mannose appearing because you can't process it, but it's certainly available for recognition. And just to show that in more detail, in cyan is the enzyme docked on HIV, and you can see in green is its substrate, and it entirely envelopes the surrounding glycans. So this is one of the mechanisms in which mannose appears. It's a steric blockade. So on the SARS-CoV-2 spike, we were able to collaborate with Jason McClellan's group in the University of Texas, Austin, and they provided an initial batch of protein, but also the expression vector. Now, this is for the later strain, SARS-CoV-2, and the key thing is here it's stabilized. So it forms, and they use the same trick that many people in the HIV field and others have used, of this proline stabilizing mutations. Now, this sort of traps it in the pre-fusion state, and in our hands it behaves really well. You can see a nice gel band, predominantly trimer, and we size exclusion purified it. Oh, I should say, not only did we have the batch from Texas, we also made it in Southampton as well, 2 additional batches, and we did our LCMS profile. Now, I have a habit of simplifying glycobiology, and it's you're sort of damned if you do, damned if you don't, because you show the heterogeneity and you lose people. But actually, there's you can simplify it massively to just say oligomannose versus complex. So I'm just including this to say, yes, behind the scenes is a lot of heterogeneity. This is in the supplementary information, but we tend to simplify it down into categories to make it more understandable. And this shows the oligomannose content from the 3 replicates, site specifically, with the green showing the oligomannose and the more processed sites. Now, one thing you can immediately see is, despite the fact that different people have made batches in different countries, this is a highly stable glycosylation pattern. We're not seeing huge diversity of glycan signatures, which is really reassuring. In the HIV field, this has been considerably, well, it's only recently been achieved, getting stable, consistent immunogen development, but the coronavirus spike seems much less prone to this variation. Now, of course, we can then map, using the cryo-EM structure, the processing states across the trimer. And here we're using some categories here just to show the emphasis of the hotspots of really protected oligomannose sites, but also in orange, you'll see this intermediate level where you're getting mixed sites. So there's still quite a bit of mannose being present, but of course, it's part of the heterogeneity. And just like in HIV, the areas which show slightly more impeded processing tend to cluster together, but it is definitely neighboring protein effects and also neighboring glycan effects that lead to that. Now, we can obviously use all the data to plot on a variety of different signatures, and here motivated by the fact that fucose is targeted by this antibody. I've shown a heat plot of the fucose, and sure enough, in the recombinant system, we're getting a nice level of fucosylation at that site. Now, this is also quite a good teaching point, because with the coronavirus budding pathway, it buds from the ER, you might be worried about the glycosylation being vastly different and maybe dominated by mannose. But of course, the presence of antibodies, neutralizing antibodies that target fucose, does reassure you that processed material is very much a signature of the actual virus, as well as more structurally defined oligomannose regions. and you can put them side by side. And here I've used the heat plot in green to show mannose, of course red fucose and magenta and sialic acid. And this heat plot does show rather more generously the mannose distribution compared to the strict cutoffs in the earlier slides at the base of the viral spike. And of course, if you allow your eye to flick between the two, there's a reciprocal nature. The areas which have less mannose, you get more modifications like fucose and silation. And now this shows them side by side, and really that's just emphasising that point that these, very much colourful sort of threshold colorings really, whilst they convey a lot of the information of the processing state, this sliding scale really illustrates just how extensive mannose is. And in this sense, the coronavirus is deviating away from your typical host proteins. Really interesting comparison though of these glycan shells. Now this is some wonderful work from Zach at Scripps working with Andrew Ward, and he's been looking using cryo-EM at the interaction of the glycans forming these sort of glycan clusters. And I love making comparison across these different viral classes, and you'll see that SARS is not exhibiting the same level of clustering. As you get these isolated stems, but it's quite disordered. So what we see is HIV very much on the extreme end of clustering, and SARS are really less so. So there's less mannose and less actual structural organisation of the glycans. And when you put them all side by side, showing the amount of mannose, you'll see that the SARS and MERS cluster at one end of the level of oligomannose, and with HIV and Lassa fever very much more oligomannosilated at the other extreme. So that illustrates there really are quite considerable vulnerabilities in the glycan shield, and you can actually plot the sort of density, a measure of the density of the glycans versus the measure of the oligomannose, and you can see a nice correlation with the coronaviruses. And HKU1 is another coronavirus clustering at the bottom, with SIV and Lassa fever showing much more oligomannose content. Now, what about O-links? Now, our experience with O-links from HIV is often where people are seeing them. It can be real, but it can also be an indicator of aberrantly folded material, or it's just an artifact of fragments. And we see this not just in viral proteins, but also in mammalian proteins. If you're studying things outside of that oligomeric context, you get much more O-links. And this is just one example where we see, say, about 5 or 10% O-link glycosylation in a region on GP120, and this pretty much disappears as you moved into the trimeric form, and in particular the native-like trimer is almost not measurable. And so when we look at regions where people are reporting O-links, we see, and this is one such region, the 323, 325, it is massively dominated. So what's 99.8% unmodified, and I've included some rather ropey spectra showing what we probably think are legitimate O-link series, representing a total of 0.2%. But you can see the evidence for these is much weaker, and it's predominantly unoccupied. So that really is an important feature. Again, N-links can be used to look at the immunogen integrity, but likewise O-links are a really important signal. So on that, I'd like to just highlight our funders, the NIH, Melinda Gates Foundation, IRV, and in particular the people who've done the SARS work here highlighted. This is Yasunori controlling the mass spec, with Joel Allen in the background doing appropriate social distancing here. So on that, I will sign off and happy to take any questions.
Fantastic, thank you ever so much, Max. Yes, so with regards to questions, because there's a short delay between us and the actual showing of this, basically if anyone asks a question as this talk ends, it'll take a couple of moments just for it to come through. So I'll just remind everyone, if they do want to go over onto the Slack channel, there is that option there to put those questions forwards, and please react to any questions that you like the sound of and you'd like to hear the answers to. Okay, so the first question we have coming through is from Harry. What cells was the S protein expressed in, and would this be expected to be different depending on cell type? And would this be true in vivo also if the virus infects different cell types?
So firstly, the protein was expressed transiently in 293F cells. All our lessons, really, because we're obviously everybody's new with this particular coronavirus strain, have come from HIV, and there you can express, and we've done comparisons between CHO in stable cells and transient in 293T, and what's really interesting is the mannose content is very stable between the two. And this is because of that steric basis. The enzymes just don't work. And because the monosidase enzymes are so well conserved, they really, all these different cells seem to exhibit that same behavior. Now, in terms of the precise nature of the complex glycans, that can change wildly and is heavily dependent on the cell line you're producing. And that's one of the motivations when I show this, and to simplify some of the complex processing, because it's less informative, it's much more connected with the experimental setup you have. Now, this mannose lessons, the amount of mannose there is, we can also look and compare, say, to virions. Again, this is just HIV specific. The lesson there is that sites which are very dominated by mannose remain dominated by mannose, and where you have those mixed sites, the ones I showed in orange, where there's a bit of mannose, a bit of complex, because we think the protein is membrane tethered and there's generally slower secretion, I think, those tend to go a bit more to completeness. You get slightly less mixed sites and much more extreme complex versus mannose. In coronavirus, there was a lovely study by Rich et al. About coming up for 20 years ago, where they looked at just mass spec of coronavirus derived material, and that showed about 30% mannose, 70% processed. So strangely, it does seem to be illustrating that the recombinant system is capturing roughly what has been observed previously in coronavirus. So we can all be braced for big shocks, and there's lots of ways that viruses mess up cells. So we have to be very open to a wide range of budding processes in actual virus infection. But in experimental systems, they do seem to agree pretty well.
Fantastic, thanks. Another question we've got come through is asking, are there any examples of biomarkers for identifying coronavirus? And are these glycosyl chains fixed composition, or do they too keep changing?
Well, I think I can't really comment on the biomarker in terms of self molecules. Others would be better placed to comment on that. But in terms of the actual viral detection of viruses, I think the next speaker is going to be presenting on that very topic. So I will again cede ground to the experts on that.
Fair enough. Frank has said, great work, Max. Is there a chance that elements of the glycan shield itself can be antigens for vaccine development?
Yes, in fact, hence my slide showing the recognition of an antibody against the fucose containing, an epitope displaying fucose. So yes, there's a chance that those antibodies can be elicited, but the big warning is that in HIV, it's really hard to elicit these mixed protein sugar epitopes. So my hunch is the vast majority will come out as being protein directed, and trying to tease your immune system to incorporate glycans may be quite an unnecessary challenge, but like that. So I think a good focus on high quality protein components with an eye on the sugars is probably the way to go.
Great. So next question we've got is the glycosylation pattern indicates it is easier to develop a vaccine, correct? Where do you expect an effective vaccine will be developed? Sorry, when do you expect it?
Oh, when?
Yes. Yeah.
I don't think I should be pontificating on precise timelines, mainly because the there's so many sort of non-scientific aspects to this to deliver an effective vaccine, so it's a very difficult one to answer. But yeah, the low density of shielding is quite encouraging. When you look at, say, Lassa virus, that really has, it's a little ball covered in sugars, and that, by all accounts, has got you get quite low titers against that, and I just don't think we're going to have the same type of barriers here. But of course, that's very much a glycan-based view, and there are lots of other parameters feeding into just how good an immune response will be.
Okay, well, one quick final question. Can glycation process affect the ability of virulence of COVID-19?
I think so. There's lots of, so by glycation here, we're meaning glycosylation rather than the sort of dietary modifications. But with glycosylation, I think the role of complement activation will be very interesting. So the degree of inflammation seems to be really quite a notable feature of the disease. And all these viruses are capable of activating complement. The sheer density in coronaviruses of the spikes, remember HIV, you'll get, say, 10 spikes here, you've got the much higher density. So I'd envisage that the glycans will be quite an important, or have a sort of role in the pathobiology, and there could be many mechanisms, but immediately you can see it as a target for complement.
Brilliant. Really, really lovely stuff, Max. Thank you so much for your time.
Well, wonderful, and thanks for the questions.
Okay, so next we'll be handing over to Andrea, Professor Andrea Sinz, from the Department of Pharmaceutical Chemistry and Bioanalytics in Martin Luther University in Germany. Andrea's lab focuses on the development of novel tools and workflows to promote the cross-linking mass spectrometry approach, a technique that has become immensely popular in the past years to study protein 3D structure and to delineate protein-protein interactions. Andrea said that she'd very much like to have a discussion and as many questions as possible out of this talk, so please feel free to ask any during or throughout as we go on Slack. And with that, I will hand over to you, Andrea. Thank you very much for being part with us today.
Thank you very much, Harry, for inviting me to give this webinar. This is really exciting. And close to me here in a proper distance of 6 feet is Christian Ehling, who is one of the co-authors of this study. So this is in fact only a very short work we did, because as you said, usually we're working with cross-linking mass spectrometry. But today I would like to share this simple study, I would say, on the mass spectrometric identification of SARS-CoV-2 proteins from gargle solution samples of COVID-19 patients. So we performed this study at the beginning of April, and this is our group. As I said, we're usually working with cross-linking mass spectrometry, and these are the 3 people who were mainly involved, Christian Ehling, the Tensler, and I was only the one then writing it up. So the cross-linking we're doing usually is going to delineate protein conformations and also protein-protein interactions, not only on purified proteins, but also mapping interactions in cells and also in tissue. And this has been sparked, of course, by the instrumental development in mass spectrometry and also by the development of novel reagents. And this is what we usually do in my group. Just to give you an overview and tell you that we're not virologists or usually not working with viral proteins. So the goal of this study I'm presenting today is we just wanted to quickly check whether we can use mass spectrometry to detect SARS-CoV-2 proteins directly from gargle solutions of COVID-19 patients, because these gargle solutions were the samples we got from the clinics, and we thought it's pretty easy if we can use mass spectrometry as a tool for a very quick detection. And of course, with the goal later on to develop that into a routine diagnostics, but that's far away, this goal, let me put that straight. So the steps of our protocol are really super simple. We first performed an acetone precipitations of proteins contained in the gargle solution. Then we performed tryptic digestion of the proteins contained in solution and performed the targeted mass spectrometric analysis, just to give it a first try. And in fact, we were quite surprised that it really worked. We could detect some virus proteins in there. So this is just the overview, but I think I don't have to go so much in detail after we heard from Max already about the glycan shield. So we have the spike glycoprotein as a trimer, then the nucleoprotein, N protein, together with the RNA inside here, and then the membrane protein M, and then the envelope small membrane protein pentomer E. So I think that is known to everyone. So what did we do now? We took the gargle solutions and we had 20 milliliter gargle solutions in 0.9% sodium chloride that the patients were using to gargle like 10 seconds. So that's how the samples were obtained. And we only had 3 patients. So this is where we got access to, and all of these patients had a confirmed COVID-19 infection. And our TQ-PCR analysis had identified the E, S, and N gene RNAs from SARS-CoV-2. And I have to also say that the N protein, the nucleoprotein, so the gene of the nucleoprotein is that is mostly or mainly expressed in the COVID-19 patients, in the SARS-CoV-2. So after we got the solution, we took only 750 microliter out of these 20 milliliter of the gargle solution and precipitated that in 1 milliliter of acetone at -20 degrees, and then stored everything overnight at -20 degrees. So only a very small fraction of the whole solution was taken here for mass spec analysis. The analytical workflow is as follows. So we centrifuge the acetone precipitate first, then remove the supernatant and only took the pellet. And then we work the usual proteomics workflow. So we dissolve the pellet in smart digest buffer, just put it to 75 degrees, 10 minutes. Then we removed the glycans by adding PNGASF, 50 degrees, 10 minutes. And then we added the smart digest trypsin, which did the digestion and also reduction alkylation. And then we performed nano HPLC MS-MS with an ultimate RSLC 3000 system coupled to an Orbitrap fusion trihybrid mass spectrometer. And the LC conditions are shown here. So first we did the trapping of the peptides with a PepMap C18 column, just trapping the peptides at 15 minutes 0.1% TFA, and the separation was done with a system from Pharmafluidics with a 200 centimeter system, the MyPack system C18, and we worked with a very long gradient that in total is 180 minutes at a flow rate of 300 nanoliter per minute. So you can tell now, of course, when you want to use this method in the routine diagnosis for SARS-CoV-2 to detect in COVID-19 patients, this gradient, of course, has to be improved. But as I said, this was just an initial study to check whether we were able to see peptides from the virus in the gargle solution. So this clearly has to be optimized. So this is the usual gradient we use for our cross-linking mass spectrometry samples. Okay. So the MS conditions are shown here. So we use an inclusion list of tryptic peptides from the SARS-CoV-2 proteins, and if none of the viral peptides were detected, a top 5-second method was applied. MS-MS was performed in parallel acquisition mode, Orbitrap HCD and linear ion trap CID MS-MS. And for doing the database search, we searched the UniProt human database and then the SARS-CoV-2 database. At that time, when we did this analysis at the beginning of April, the SARS-CoV-2 protein was still not included in the UniProt. So we had to use this side. Now it's included, but I just show you the slide later. Then data analysis was performed with the Proteome Discoverer, and whoever wants to check the raw data, I think this is really important that you can go in the data yourself and check the data quality, that all the peptide matches are really high quality. You can check for yourself. They're all deposited in the PRIDE at these accession numbers shown here. So please feel free to reanalyze the data and send me your comments then. So this is the proteins of the SARS-CoV-2 database which we were using here, because at that point it was not added just in the UniProt database, so we had to add it extra. And now the outcome of the analysis. So we had 3 patients. One patient had the lowest virus load according to the PCR analysis, and in that sample we did not detect any virus proteins. So you can see here from the list of identified proteins that these are the usual proteins you would expect in saliva and also in the throat and the mouth of patients. But the next samples, patient 2, you could see we were really surprised, I have to say, that we identified 4 unique peptides, 15 peptide spectral matches from the nucleoprotein of the SARS-CoV-2. And the third sample, the third patient, we could detect 6 unique peptides with 12 peptide spectral matches in the solution. And this was quite yeah, we got quite enthusiastic about that, because with this simple approach that we could really identify peptides in the 2 patients that had a higher viral load than this other patient. So it shows that the method in principle is working. Of course, it has to be improved still. And now to take a look what these peptides are, take a look at the sequence coverage. So the peptides that we found are shown in green here, and this peptide from amino acid 41 to 61 is also highlighted in gray. This is a peptide we found in both patient samples, while the other peptides were found only in one of the patient samples. So the next thing is now to check whether these peptides are really unique for SARS-CoV-2, or if they are also present in some other organisms. But just to give you an idea now about the quality of the sequencing data, MS data is shown just an example. This spectrum, fragment ion mass spectrum of the 4+ charge signal of this peptide we found in both samples from amino acids 41 to 61, and it shows that we have a pretty decent line of B and Y type ions, and so the match looks really good. And as I said before, all raw data are deposited in PRIDE repository, so you can check them yourself and reanalyze them. Then we performed a blast search to see whether the peptides are really unique to the SARS-CoV-2, or whether they're also found in other species maybe. And as you see here, this first peptide 41 to 61 here, that is was found in both samples. This is also found in the SARS-CoV virus from 2003, and then also in the bat coronavirus, while the other peptides we found are all unique to the SARS-CoV-2. So they are not found even in this SARS-CoV virus and in the bat coronavirus, as you can see here. So they are unique, and they can be used for diagnosis by mass spectrometry, as you can show here. This is also the other peptides that were found. So that's already the end of this brief story. So we were able to identify unique peptides originating from SARS-CoV-2 nucleoprotein in gargle solution samples from COVID-19 patients. And of course, the method has to be improved regarding analysis time and sensitivity. So we're currently working on that, but of course it's really problematic to get patient samples. There are strong safety regulations, and we were only allowed to get the samples, the acetone precipitates already, to work in our lab. And the future goal is to develop this method into a diagnostic tool of COVID-19 patients that might at some point complement PCR methods. So I think mass spectrometry can really contribute here really nicely. Okay, that's the end of my talk. That's the group again. You see in the background, you see the beautiful city of Erfurt. And thanks again for inviting me to share this, I would say, very initial data. And I'm looking forward to questions now. Thanks.
Fantastic. Thank you so much, Andrea. We've got lots of questions coming in. I will try and go through a selection of them. So we've got quite a few relating to the acetone precipitation process. So Harry's asked, have you done shorter acetone precipitations? Overnight is potentially a long bottleneck in the analysis time.
Yeah.
Or have you been able to test other precipitate methods to improve sensitivity?
Yes, so actually, as I said, this was just the first try and we were ourselves really surprised it worked that well. And we did the acetone precipitation because we have in the past tried different precipitation methods and acetone just worked best. So that's what we started with. And overnight was just in that workflow what we did it. But when you add the -20 acetone, of course, you can shorten that part, I would say.
Okay. So another question relating again to the precipitation. When you precipitate with acetone at -20, do you find you're precipitating all protein and not specifically enriching for virion content? Have you varied temperature and ratio of acetone to sample to see if a difference in virion enrichment over other proteins is found?
So I present what we found. Of course, you precipitate, you saw what we discovered in our analysis. So these were mainly human proteins, what you find, of course, in the mouth and in the throat and saliva of patients, of humans. And at that point, as I said, we only had these 3 samples and this very limited amount of material because we could not go back now to the clinics and get more samples out there. And we did not try at the moment other precipitation methods. We just gave it one try and that's the outcome. So there is a lot of room to try and check now, and that will be the next steps. Yes.
Fair enough, yeah. Someone has asked, do you deactivate the virus before the precipitation?
No.
Okay. Fair enough. So for your goal of developing this as a diagnostic tool, what MS platform would you choose for that purpose?
Okay, that's a good question, but I hope not from a vendor. So, of course, I would now go for triple quad instrument because to improve the sensitivity and then also improve the LC part of the method, maybe go away from nanoflow to cup flow or also normal, we don't know yet, but then just shorten the gradient and then use a triple quad mass spectrometer. I think that's the next steps we should take.
Great stuff. Do you think that in addition to a diagnostic tool, this also has prognostic value, such as predicting patient trajectories and outcomes of the disease, etc.?
This is a difficult question. So as I said, I'm not a virologist. And you know that the virus, when you can detect them in the gargle solution, it's only an acute phase marker, right? Because then the virus apparently goes down to the lungs and then it's not detectable anymore. So what we show here is just an acute phase marker in that way. And about prognostic marker, I cannot tell much because that, I think that's what the question means, how severe a disease is then progressing. I don't know if that's what the people who asked, the person who asked the question was referring to. But of course,
follow it up if needs be.
Yeah. Of course, then you would also aim to quantify, and that's also a good question because the virus load that was determined, but I can only give a very rough estimation of virus load in these patients, and according to the people from the clinics where we got the samples, it is like between 10 to the 6th to 10 to the 8th per microliter of virus particles. So it's a very high virus load in these 2 positive patients. But about prognostic markers, I cannot say anything.
Out of interest, have you compared this data to any healthy gargle solutions to see if any proteome changes are happening that might be immune related, that are independent of the virus proteins?
We have not done that yet. As I said, this is very initial, very initial stage at the moment, and we have not compared, but that's a point I've already thought about already, just proteome changes induced by the infection, whether we could detect that. No.
Do you know what happened in terms of clinical outcome to the 3 patients that were measured?
Two of them got home healthy, and the other one was in the clinic, but then was also released.
Good to hear. After optimization that you mentioned, how long do you envisage the cycle time? Will the complexity of this MS method pose a challenge for using it as a diagnostic tool?
What do you mean? I'm not sure if I understand the question correctly. You mean the MS measurement itself, or what?
I think they're referring to kind of the amount of time it'll take and the complexity of actually preparing the samples.
I think it's not so difficult actually, because what you have to do is just do the precipitation, right? And then if you the digestion can be reduced also, I think, to a short time. That takes, of course, time. But then the analysis time itself, the LC-MS, that can be really shortened a lot. And this has to be shortened, that you can go down to very short gradients, maybe 15 minutes or 10 minutes even. And then it's not a really, I think the labs who do clinical diagnostics, who already have triple quads in their labs, I think they should be able to do it. Or do you compare it to MALDI-TOF measurements? Is that what you refer to probably when you just do like a pattern recognition or something, a biotyping? That's of course an alternative approach then.
I believe we'll have talks on a fair bit of that in the next time.
Yeah, I saw that. It's good. It's interesting. Mm-hmm.
Did you search for any post-translational modifications to the viral proteome?
No, we did not. So we removed the glycosylation, and then of course we checked for modifications like methionine oxidation, then of course carbamidomethylation, the usual things, but not on other modifications.
Next up is a question from Mark. Can you find the SARS peptide in sample 1 if you look at the extracted ion chromatogram?
We did not, I don't know, we haven't done it yet, so that is something we can explore still, yeah.
What do you think cross-linking mass spectrometry can be used in conjunction with cryo-EM to better understand the structure of the viral proteins?
Yes, that's maybe a future project. We don't know yet, but could be. Mm-hmm. So because the cross-linking will give you a 3D structure information on the proteins itself, and this could be used, of course. But as I said, we just jumped into this project, never worked with virus proteins before, so it's all it's a new field for us too. So
following up from a few questions previous, what do you think, I mean, how quickly do you think the actual cycle time can be complete for analyzing per sample?
How quick it can be?
Mm.
You mean the whole analysis from precipitation, taking out the gargle solution and then get the result?
I think probably just in terms of the mass spectrometry time.
Yeah, but that's what I was saying. So I think when you shorten the gradients to very slow, very short times, that you have like only 10 or 15 minutes gradient, and then you should. That would be the limiting factor of your LC-MS method then, of course. But the whole analysis, I don't know how low you could go, maybe 1 hour for the whole thing or so, maybe 2 hours, something like this, in that range.
What are your next steps? Are you continuing to optimize the mass spec? What have you got planned with the rest of the gargle solution?
The rest of the gargle solution is in the clinics, and we don't get hold of it at the moment anymore. But we hope to get new patient samples soon. And then the next step will be, as I said, that we go to a more sensitive mass spectrometer and then optimize also the LC gradients.
Okay. Are you considering the use of anything like SP3 as a kind of alternative and potentially to gain a higher precipitation rate? than acetone?
We're now exploring, it's also a good question, different precipitation methods, but of course there you have to get hold of material. You can optimize that with healthy samples or just gargle solutions, optimize that. Yes, this is something we explore, but I think the main focus should really be on optimizing the LCMS method, not the precipitation. I don't see a real problem there. I think the acetone precipitation worked pretty well.
Fantastic. I think we've pretty much come to the end of our hour. Thank you ever so much, Andrea, for answering all those questions.
You're very welcome.
Your assistant.
My pleasure to participate. Thank you.
Lovely. And thank you ever so much for everyone for joining us today. Before everyone disappears, we do have a couple of quick things to put up at the end. I'm not sure if you can advance that for us, Harry.